Okay, so here we are with part 3. Now this one gets pretty complicated. Allow enough time to talk about or to study about capillaries. So we're going to start by going back to the beginning of the chapter and look at the structure of capillaries and capillary beds, and then we're going to see how nutrient waste exchange in capillaries actually occurs. Right? So reviewing the structure of capillaries, they're very small. They have the thinnest walls. They have only an endothelial layer. But they have a lot of blood flow. Remember how high their total cumulative cross-surface area was. So the total capillary flow from one moment to the next is about equal to the cardiac output. And like all parts of the circulatory system, pressure and resistance are going to determine flow. So in terms of structure, just an endothelium with a thin basement membrane. They cannot vasodilate or constrict. The only control over them is at the precapillary sphincter. We divide them into 3 kinds. The most abundant kind is the continuous capillaries found in muscle, connective tissue, specialized continuous capillaries in the central nervous system and the thymus. They are very basic, and we'll look at them most particularly. The reason they're not found in epithelia and cartilage, remember, is that those 2 kinds of tissues are avascular. They have no blood vessels of their own. Similarly, you wouldn't find them in the cornea or lens of the eye. A special kind of capillary is called a fenestrated capillary. These have an abundance of little pores, openings, or thin spots in the wall, and that's wherever you have to get just particularly larger solutes going very rapidly either into the capillaries or out of the capillaries. So they're found wherever fluids are going to filter in abundance, like the choroid plexus, which you remember in the ventricles of the brain and forms cerebrospinal fluid, endocrine organs that are secreting protein hormones, kidneys, and we'll study those specifically when we get to the kidneys, and in the intestinal tract where you're absorbing a lot of nutrients very quickly. Our third kind of capillary is called a sinusoid or sinusoidal capillary. These have huge gaps between the adjacent endothelial cells. Sometimes they're called discontinuous capillaries because of that. And these huge gaps permit free exchange between the interstitial fluid of the tissues and the plasma. It's like there's no boundaries, no barriers at all. So you find them in liver where you're pouring plasma proteins into the plasma of the blood. In the spleen, where even red blood cells can get out of the blood and kind of hang around the spleen, so your spleen is a storage organ for red blood cells. Bone marrow, of course, because you're forming new cells there, and so they need to easily enter the blood. And again, certain endocrine organs that are secreting very large protein hormones, and you do not need to know which endocrine organs have which kind of capillary. Okay, so here is a diagram on the left of a continuous capillary. You can see the adjacent endothelial cells have very, very tiny boundaries between them. Look in the lower figure, and you can see that little boundary between the endothelial cells, just the tiniest little slit. And these slits are called filtration slits. Endosomes and correspondingly exosomes are going to allow exocytosis of proteins to get either in or out in certain cases. On the right, you see your fenestrated capillary, and here you see that in the fenestrated capillary, you've got these little pores, which are just thin spots in the capillary wall. And then on the far right, the sinusoids shown very differently. But you can see big gaps between the adjacent cells, and you can see that they have a much larger diameter. So we'll just skip through all these. Y'all study them in more detail. Okay, capillary beds. In a capillary bed, you've got the supplying arteriole, the metarteriole, which is the part of the arteriole where the capillaries are going to arise. Each true capillary is marked by its own precapillary sphincter that guards the entry to that capillary. The thoroughfare channel is a direct pipeline between the arteriole and the venule, and if all the precap sphincters are closed, then the blood just gets shunted straight from arteriole to venule. And then, in fact, you have a bypass. At the bottom of the picture, the arteriovenous anastomosis simply bypasses the capillary bed completely. All right, so there's just a lovely diagram showing, or a photomicrograph showing you all the capillaries coming off a specific arteriole. Okay, so we're going to move to capillary exchange, and here we're going to look at the processes by which materials move across the capillary walls. Diffusion, of course, you know, is the movement along a concentration gradient. So you have to have a high concentration and a low concentration. You're going to do diffusion to get CO2 out of the blood and carbon dioxide into the blood. Filtration, you remember, is just pushing fluid through a filter from at high pressure. So the blood pressure in the capillary pushes pushes plasma through those little filtration slits in the capillary wall or through those little fenestrated pores and allows fluid volume to move in bulk flow. So fluid literally leaves the blood, and then most of it is going to be reabsorbed by the same process, sort of filtration in reverse, as we're going to see. So diffusion, remember, requires a concentration gradient, and you're going to see mostly diffusion of the lipid-soluble materials such as oxygen and CO2. All these other things that are moving are probably moving not so much through diffusion as they are through filtration, because filtration is just going to carry the water and anything that's dissolved in the water with it. So it's not necessarily moving across a concentration gradient. It's driven by hydrostatic pressure, and that's going to be the bulk of most of the water and solutes forced through the capillary wall and leaving behind in the blood the plasma proteins. And the plasma proteins are going to create an osmotic pressure that drives reabsorption. So filtration pushes fluid out. Osmotic pressure, osmosis, pulls the fluid back in. And so we need to understand the balance between these pressures, the hydrostatic pressure, the blood pressure pushing it out, and the osmotic pressure of the blood colloid osmotic pressure due to the plasma proteins, which kind of pulls the fluid back in again. All right, so this diagram shows you that capillary hydrostatic pressure pushing against the little filtration slits, the plasma proteins in the blood, and almost no proteins in the interstitial fluid. And so those plasma proteins stay in the blood. They're too big to filter out, but water and the small solutes can go right through those filtration slits. So it's that interplay between filtration and reabsorption that balances out the fluid leaving through filtration, the reabsorption due to the osmotic pressure, and that filtration carries nutrients with the water out to the tissue spaces, and that reabsorption process pulls in the waste products and perhaps anything that doesn't belong there, whether it's bacterial toxins or chemical signals or whatever. So the two primary forces that affect filtration and reabsorption are the capillary hydrostatic pressure and the blood colloid osmotic pressure. And the hydrostatic pressure is offset by the interstitial fluid, which also has its own hydrostatic pressure. For interstitial fluid, it can actually have a negative pressure because sometimes that fluid is getting sucked into the lymph capillaries, or it can be a positive pressure in places like where tissues can't expand, like the soft tissues under your fingernails, or the chambers of the heart, because the pericardial sac is pretty rigid and doesn't let things expand. All right, likewise, interstitial fluid has a colloid osmotic pressure. Now remember, the colloid osmotic pressure is due to proteins, but there aren't very many proteins in interstitial fluid. So the interstitial colloid osmotic pressure is low to non-existent. All right, so the net filtration pressure is the difference between the hydrostatic pressures and the osmotic pressures. This formula is important to understand. Those four factors involved in that formula. It's important to understand why fluids will accumulate in the tissue spaces. That's the puffiness associated with edema, or why you're going to pull extra fluid out of the tissue spaces and into the blood. So a lot of that's going to depend on those four forces. And so this gets us to that overall idea of capillary exchange. Again, fluid at the arterial end, where the blood pressure is high, tends to filter out of the capillary to the interstitial fluid, and that, in fact, that's how interstitial fluid is formed. And then at the venous end of the capillary, fluid moves into the capillary because the blood pressure is low. The transition point, there's going to be one. We don't really care exactly when it is, but you're just going to have a shift from filtration to reabsorption. And the balance of filtration versus reabsorption is actually in favor of excess filtration, but that extra fluid is supposed to enter the lymphatic vessels. And we'll worry about what happens to that when we get to the next chapter, which will be after this test. So here is a diagram showing you the actual pressures. So looking at this figure, what you see is that the blood pressure at the arterial end is about 35. The osmotic pressure is about 25, and the blood pressure at the venous end is only about 18. So here, the difference between 35 and 25 is 10. And here, the difference between 18 and 25 is 7. And the difference between positive 10 and negative 7 is positive 3, which means you tend to filter out about, at a pressure of about plus 3 millimeters of mercury, a total of 3.6 liters a day, which is going to flow into the lymphatic tissues. All right, so things that can disrupt these things. Anything that lowers the blood volume, like hemorrhage, is going to reduce the hydrostatic pressure. That's the blood pressure. Dehydration causes you to lose water. That makes your body fluids more concentrated. That's going to increase the blood colloid osmotic pressure, so you're going to pull extra fluid from the tissue spaces and maintain your blood volume. So whether blood pressure drops or blood colloid osmotic pressure rises, either way you tend to pull fluid from the tissue spaces into the blood. If the opposite occurs, if the hydrostatic pressure rises or the colloid osmotic pressure drops, those are the things that are going to result in edema. And so you're going to see that in someone with hypertension. Pregnant women get issues with preeclampsia, and they get swollen ankles. That's edema. People with end-stage liver disease have a very low colloid osmotic pressure, and they get those big swollen bellies called ascites. All of those are examples of edema. Okay, so perfusion of a tissue means blood flow, and you've got to have adequate perfusion of tissues to maintain the health of the tissues. Without adequate perfusion, tissues can die and become necrotic. Obviously, that's bad. So adequate tissue perfusion is affected by all of the factors that affect blood flow that we've already talked about. And so the cardiovascular system works together to maintain normal blood flow, which gives you normal or health, which is, of course, homeostasis.